It's my pleasure to introduce our first speaker. He's world-renowned for his research on mercury and its relationship to human neurological diseases. I first met him at my first Academy meeting in 1996, and he's changed my life ever since. He has a master's in chemistry from the University of Idaho, a PhD in chemistry and biochemistry from Washington State University, and served as chairman of the Department of Biochemistry at the University of Kentucky from 1996 to 2005. He has been published, has lectured worldwide, and testified before Congress on the dangers of mercury exposure, and is currently dedicated to the development of Erminix. It's a safe key leader for mercury, lead, and other toxic heavy metals. Please welcome to the stage Dr. Boyd Haley.
Thank you, Dan. Good morning, everybody. I've only done this for 40 years, so you would think, there it goes. And this is the talk we're talking about. And so this is the first part of it. The two identifiable groups most susceptible to COVID-19 infection and death are those with underlying medical issues and the elderly, over 60 years of age, which is me and a whole bunch of the others there. And I would tell you, I've been taking this compound since 2006, 14 years. And in those 14 years, I have not had one viral infection, not one cold, not one flu, and I used to get them all the time because some of my friends here will attest to, I'm an avid duck hunter. I go out in the worst of weather early and I get sick. Okay, so the one thing, and if I watch them talk on television, they ask our experts that Trump brings on every 2 weeks or every week or every day for a while, and they ask him, why is it that certain people get the virus but they don't get sick? Well, it's because they're young. Well, the virus doesn't know your age. There's chemistry behind it, and that's what drives me crazy. These people should be looking at what's the chemical difference between an older person and a younger person that gets the infection but doesn't get sick. And it's very well known. I mean, there are differences that happen, and one of them is your glutathione levels go in the toilet when you get over 60. They go down very fast. And glutathione is a major detox system product in the body, gets rid of a lot of toxins. Your body has the P450 system that has glutathione S transferase as a key enzyme, attaches glutathione to molecules that build up in your body, and when they get glutathione attached to them, they become water soluble, and they're marked for excretion from the body through the biliary transport process. Mercury being one of those, and compounds even like my compound, once it binds and chelates mercury, it just sits there, it's not toxic. But the body gets rid of it, and it takes it out through the fecal route, and it takes it out probably with glutathione attached to it. So glutathione is one of the most natural antiviral molecules in the cell cytoplasm where the viruses are replicated. And I'm going to show you the data that's not new data, that's been around a long time. But we propose that a very effective method of accomplishing increased glutathione would be the ingestion of a drug supplement in development that has been shown to increase glutathione levels in patients suffering from oxidative stress. So the first part of this presentation, I will show you the data that's out there, not done by me, done by virologists, showing that glutathione is the best inhibitor of viruses known. And how they do this, they have a planchet, they're growing cells in there that are affected by the virus, they add the virus, and then they add drugs that inhibit the viral replication or to test it. And the thing that they can add that is absolutely without toxicity and totally prevents the viral replication is glutathione. So there is a tremendous similarity. Our compound causes glutathione to go up in the body. It also mimics glutathione. If you look at the structure of our compound, it looks like two glutathione molecules attached together. I mean, it was engineered to mimic the redox system of the body and to also bind and remove heavy metals, doing it better than glutathione. And so we propose that the use of this antioxidant chelator would be an effective treatment and preventative for COVID-19. Actually for any viral infection. And the first consideration, does reduced glutathione have a role in eliminating viral infection? It does not have a role. It has the major role. And they're trying to tell you that it's the production of antibodies that gets rid of most of the virus. I'm telling you, getting rid of a virus is detoxing, and glutathione plays a major role. If you don't have it, no matter how much your immune system is working, it is dramatically weakened because glutathione attachment is a major part of the immune system. So here's the studies, and look, I could sit up here and give a talk, an hour talk, on any one of these things, but we're going to go through it. So let's read the highlights of what the experts say this says, because if I try to go through each one of these in detail, I'd never get through the talk. And this is, the title says all about this. Glutathione inhibits HIV replication by acting at late stages of the virus life cycle. And we'll talk about that. And they're talking about the disulfide bonds on the coat protein. Glutathione is inserted in there with glutathione S transferase. So when the virus made in your body and you're healthy, what happens inside the cytoplasm where glutathione levels are very high, there's an enzyme called glutathione S transferase. It transfers glutathione onto the virus coat protein by exchanging it with the disulfide linkages in the coat protein. So that virus leaves your body coated with glutathione. That does two things. It keeps it from binding to the next host cell it runs into. It slows that down. And when it goes through the biliary transport system in the liver, there's recognizable sites in there that bind anything that has the glutathione tail hanging out on it, and it puts it into the bile, and you defecate it out of your body. So that's how glutathione gets rid of the virus. And how you get rid of the virus, it's not by an antibody binding it and carrying it out. I mean, that happens, but I don't think it's the major way. Okay, this thing says the overall data suggests that glutathione can interfere in the late stages of viral replication, and it does this by binding into the coat protein. This would be in agreement with the data obtained in cells exposed to herpes virus type 1, a DNA virus, or Sendai, an RNA virus, showing that the suppression of viral replication by GSH is related to the selective inhibition of the envelope protein glycoproteins. I mean, it doesn't interfere with DNA and RNA synthesis at all, but it takes the whole package and gets it out of your body, detoxifying. So, and this is the second paper, and look at the dates on these paper. Oh, it should go back. If you look at this paper, this was done in 1996. This was the paper that had me give this talk in 2011. It was one of the papers I found that I thought was very important about the role of oxidative stress, because glutathione levels, when you're under oxidative stress, your glutathione levels drop like a rocket. I mean, they go very low. If you get old people have about a level of 200 units of glutathione versus teenagers who will have around 2,000. So when you're young, you have very high levels. As you age, especially when you get past 50 or 60, and depending upon your health, your glutathione levels start going down. And so this paper here, again, 1997, glutathione deficiency is associated with impaired survival in HIV, AIDS disease. And I guess this says, it argues strongly that the unnecessary or excessive use of acetaminophen or Tylenol, an FDA-approved drug. In case you're wondering, about 500 people a year die of Tylenol toxicity. And Tylenol kills you by lowering your liver glutathione levels dramatically, because glutathione binds to some of the products of the P450 system that modifies acetaminophen, and it takes your glutathione levels down dramatically, and you die because of very, very low glutathione, and you cannot suppress the damage caused by oxidative stress. So it plays a very important role in this. And so it says that, you should not be taking Tylenol. And I just talked to someone today that was talking about a sick person, and they're taking Tylenol because they have a headache. Tylenol is not a good product. It shouldn't even be allowed. But the FDA, if they approve something, they keep it forever. I mean, it causes many people to have problems they shouldn't have because they're already low in glutathione from oxidative stress, and so we're giving them a painkiller that lowers it and sends them to the hospital, and hopefully they survive. Okay, the next one is the molecular mechanism of decreased glutathione content in HIV virus type 1 TAT transgenic mice. And the HIV virus, they have a hard time getting rid of it. Back when this was done, and this was published in 2000, they were finding out that they didn't have enough glutathione and they couldn't get the virus to go away. And that's because it has the TAT. And you read that thing here, TAT is a transactivator encoded by the HIV genome. It is sufficient to cause GSH, or glutathione depletion in vitro, and is implicated in AIDS-associated Kaposi's sarcoma and B-cell lymphoma. In other words, there's a protein that's encoded by the virus that goes in and lowers your glutathione levels such that the glutathione can't be used to get rid of the virus. And you can't get, this is a very difficult virus to get rid of and get over because of its its ability to prevent you from having good glutathione levels. I mean, gay people should be very much advised not to take Tylenol. I mean, if you're going to be in this associated system, therefore, I mean, I guess it's all summarized in this one thing. Therefore, TAT, which is a transactivator protein, it causes glutathione depletion, appears to cause decreased glutathione in vivo, at least partially through the modulation of the GSH, glutathione biosynthetic enzymes. So there's another aspect of this. Here's another paper, Inhibition of Influenza Infection by Glutathione. And it says, accumulating evidence suggests that cellular redox status plays an important role in regulating viral replication and infectivity. And that says it all. But if you get down, the data suggests that thioantioxidant glutathione has an anti-influenza activity in vitro and in vivo. Oxidative stress or other conditions that deplete GS in the epithelium of the oral, nasal, and upper airway may therefore enhance susceptibility to the influenza infection. So it's all coming down, you know, anything, the virus thrives in people with oxidative stress, especially in their upper respiratory pathway. And this goes back to what came out with the COVID-19. They suggest that COVID-19 binds to red cells, depletes the hemoglobin of iron, causing free iron, which can be used to produce hydroxy-free radicals, increasing oxidative stress, and also depleting the ability of the blood to carry oxygen to cells. And these people die of respiratory deficiency. So it's all fitting in, and you've got to address this. And the people who want to sell you a lot of vaccines don't want to talk about maybe you should just take a better diet and work on getting your glutathione levels. Maybe doctors should be testing your glutathione levels, and maybe we shouldn't be recommending Tylenol and other molecules that cause oxidative stress, because a lot of the medicines that are sold to Americans induce oxidative stress. A lot of them. And so why take them? Why make people sicker by giving them something? And it says, these results suggest that interaction of core protein with mitochondria and subsequent oxidation of the glutathione pool and complex 1 inhibition. Complex 1 inhibition is on the electron transport system. Some viruses, what's in the electron transport system? A lot of iron. Mitochondria have the highest level of iron of any organelle in your body, I think. And it's in the electron transport system. You have these iron sulfur centers. And if you knock that iron off, iron is a good electron carrier. That's the reason it's good at making hydroxy-free radicals. It's a redox metal. It transfers electrons. And so with this transfer of electrons and the inhibition of that, when you stop the electron transport, you can't make ATP, you can't repair things. And at the same time, you start having the electrons coming from your TCI cycle through NADH, piling up and leaking into the body, making a lot of superoxide anion. And that, I'll show you later how that fits into this whole process. So we say this inhibition may be an important cause of the oxidative stress in chronic hepatitis C. So here's the thing. If you look at all co-proteins on all viruses that I have known of, and I'm not an expert in virology, I'm a biochemist, I'm not a virologist. But if you look at this person, that PSSP is a viral coat, and it has a lot of disulfide linkages. Yet that's chemistry biochemistry that every virologist knows, and biochemist. And then you have glutathione, which I have the red arrow on, and glutathione with the enzyme in there goes to the coat protein when it's being made in the cytoplasm, where glutathione is very high, and you have glutathione S transferase, transfer that glutathione to the coat protein. So the coat protein, which is also going to be the modality which binds to the next host cell, is now coated with glutathione. And so some people speculate that that inhibits the ability of that virus made in a cell with healthy levels of glutathione cannot go out and attach a next host molecule. For certain, This marks that molecule for being picked up by the biliary transport process in the liver, and that virus being put into the feces and excreted from the body. That's the transport process, and that's exactly what glutathione does with most toxins that it interacts with. It puts a signal molecule on it that allows it to be excreted. So the viral coprotein with a glutathione inserted in a disulfide exchange reaction marks it for removal by the biliary transport process. And again, this shows you why glutathione is the most effective inhibitor of viral replication in planchets where you control it all and nothing else is involved. I mean, it binds the coat protein, preventing it from infecting the next cell. So here's reduced glutathione, and if you look at this, there's two structures here, but if you look at the top structure, and I can't, you can't see it. You can see that yellow sulfur. That's where you insert into the protein, and the rest of this molecule is hanging out as a receptor site. There are 3 charges on it, 2 negative and 1 positive on that glutathione molecule. So it's very avidly attracted by transporters that want to bind anything with glutathione attached to them and put it into the bile. And if you look at this, remember that thiol linkage, because that thiol linkage you see there with the yellow at the top center, that's exactly what I mimicked when we made NBMI, our drug. We have 2 of those. And we did that because we wanted to mimic all the possible chemistry that glutathione did. We wanted our compound to do it also. And it does that. I mean, it scavenges hydroxy-free radicals. It binds into several molecules, causing reduction. So now let's jump forward with what the virologists are saying. This is published in 2020, this last year. Endogenous deficiency of glutathione as the most likely cause of serious manifestations and death in COVID-19 patients. And if those of you, you can go read, I think my talk on the YouTube, 2011 IAOMT, and that's what we were talking about. Glutathione is the most, levels are critical to keep from getting viral infections. All viral infections, not just COVID-19, because this wasn't even talked about at that time. And it points out that the higher rates of serious illness and death from coronavirus, COVID-19 infection among older people and those who have comorbidity suggests that age and disease-related biological processes make such individuals more susceptible to environmental stress factors, including infectious agents like coronavirus. You know, like age. No, talk about the chemistry. I mean, there are some old people, some people I've met older than me that can outrun me and live better than me and they're healthy. It's your health. What's happening? What is healthy biochemical levels of compounds in your body that are depleted in these 2 types of people? It's not their age, it's the seriousness of their illness. They are undergoing oxidative stress and they don't have glutathione to fight off the viral infection. And so if you're a medical doctor and you want to work on saving somebody you really love, make sure that you get their glutathione levels back up, and you can do a blood glutathione test very quickly. I mean, people do that. Yet medicine totally ignores that. And I remember the first time I went to a medical doctor when I was taking OSR, we called it OSR at that time, and I asked the doctor, because I was getting a physical, if he would do my blood glutathione. And he says, we don't know how to do that, we don't do that. And I said, well, why not? I said, it's associated with all illnesses. He says, because there's nothing we can do about it. That's the reason they don't test for it. They don't know how to do anything with it. That is the status of our American medicine at this time, and there's no one working hard to do it. And I think the reason they're not working hard to do it because you're interfering with the $170 billion vaccine industry if you find something to raise their glutathione levels. I mean, but that's what you're going to have to do. You're going to have to raise their glutathione levels if you want someone like that. And if you look at the response to hydroxychloroquine, you know, some people say it's absolutely beautiful for preventing the viral infection and getting over it, and other people absolutely say it doesn't work at all. The people who say it doesn't work at all are the people who are avid about getting a vaccine to recognize it. And so, you know, there's something wrong there. There's a conflict there. But look at the amount of money that's sold. There are 170 sites now that are trying to develop a vaccine. Well, how many vaccines do we need? And why are they all competing so hard? Because that's where the money is. And to find a cure for it, I've talked to several people. You cure a disease and you lose a customer, whole bunch of them. And so I think we have to get over that philosophy if we want to see people get better. And this article here is just discusses that in detail. And it says the feasibility of using glutathione as a means for treatment and prevention of COVID illness is critical. And they have to do that. But you have to learn how to get it up. And you cannot take glutathione. Glutathione has 3 charges on it. And it's made inside the cell. It's 5 millimolar inside the cell, about that level. And it's micromolar, or 1,000-fold less in your plasma. And the reason it is because, with lack of a better term, the creator made it high so it would go down the concentration gradient and carry out crap that they didn't want inside your cell, including viruses. And so when people say, well, we're going to give you glutathione IV, it's not going to hurt you, and there may be a little bit of help, but it is not going to get inside your cells and help you get rid of viruses. You have to make the glutathione inside the cell, because it's a one-way street. It goes out, it doesn't go in. And if you want to get the stuff out, it's in there with the cell. You got to have the glutathione inside. And so here's another one that came out just recently: the role of glutathione in protecting against severe inflammatory responses triggered by COVID-19. And again, this is not done in the United States. This was done in Italy. These are the people I think that have been the major pushers of this compound. And so this is a review about what I talked about. Not that they copied from me. I don't think that's true at all. But I mean, this is them trying to do what I'm trying to do, to get people aware of the fact that glutathione is critical. And it says the aim of this review is to provide a brief overview of the protective action of glutathione against the exacerbated inflammation triggered by COVID-19 and they go through the ACE, which is the angiotensin system, ACE1, ACE2 balance. Furthermore, we discussed the evidence, the low levels of glutathione found in the conditions associated with severe outcome of the disease, with the intention of looking into the relevance of restoring glutathione levels and the aim to protect the most vulnerable subjects from COVID-19. I mean, a lot of people have COVID-19 and they don't even know they're sick. And that's because they have a very good reduction. They have a very good redox system. And so this guy, this is on me, and I'm going to go through this so you show that what I said back in 2011 was true. Glutathione plays a central role in the pathophysiology of human diseases. Everybody knows that. You guys should all know that. I've talked about that so much to this academy. Glutathione imbalance is observed in a wide range of pathological conditions, including lung infections, HIV, diabetes, cancer, age-related diseases. Matter of fact, I always tell people, go to Google, type in your favorite disease, and then put a comma, and then type in oxidative stress. Oxidative stress, it's not glutathione levels, it's oxidative stress. Oxidative stress causes your glutathione levels to go down, but oxidative stress also is the hydroxy radicals are the major factor that destroys your tissue. It is the most chemically reactive agent in your body, and it destroys your DNA, your lipids, your carbohydrate, and protein. And it says, he said, if we consider conditions clinically associated with severe COVID-19 disease, we find evidence of a perturbed glutathione replenishment system. I mean, you're not making glutathione anymore. You stop making glutathione, and you have COVID-19, you're gonna go downhill. And several pieces of evidence in our biochemical suggest that low GS levels would be one of the major causes of the excessive inflammatory response. Glutathione's major function in your body, if you're healthy, is to scavenge hydroxy-free radicals that are produced by leakage of electrons from your electron transport system. That happens to all of us all the time. And you have glutathione there to make sure that the hydroxy radicals are scavenged by the glutathione, and that's what my molecule does also. That's where I designed it, with the glutathione end on it. I want to scavenge the hydroxy radicals that are floating around your body that shouldn't be there. And it doesn't do any good. There's not a need that I know of for hydroxy-free radicals except to make you get older and die. So the future clinical studies investigating levels of glutathione in COVID patients may be a starting point to explore this responsibility. Now this 2020 review is telling you we need to study this. And we knew about it in 1996. Why in the hell weren't we studying this 10 years ago or more? And here's their thing. Now this is there's nothing wrong with their hypothesis, but it's very narrow, very nearsighted. And you can see, but I wanted people to get this so they can look at it, because this is a good article. But if you look over to the right, when they put the COVID, that's the red typical COVID virus particle, you see it increases vasoconstriction, inflammation, oxidative stress, and apoptosis. Apoptosis is caused by low glutathione levels. The ratio of oxidized to reduced glutathione is what induces natural cell death, except sometimes you can make it unnatural by creating that abnormal ratio. So this is in that paper, and I suggest all of you that are interested in maybe treating people with COVID, read this, because there's no way that sitting here listening to me talk is going to get this message across to you. This is too complex, as you can see. And so in this thing here, it says, one of the subacute exposures to lead results in glutathione pool depletion and accumulation of lipid peroxidation products, and modifies the activity of the glutathione-related enzymes such as glutathione S transferase, and glucose-6-phosphate dehydrogenase. For those of you that have studied medicine, you recognize these enzymes. They're critical in reacting to disease processes. And this is from that review article, and it says, making the protein more. Glutathione can be conjugated to proteins in a process called protein S glutathionation, forming the protein SS glutathione, which is that old old slide I showed you that I produced 11 years ago. PSSG protects protein cysteines from being over-oxidized to sulfonic, and sulfonic species are not readily regenerated. Glutathione S-glutathionation also changes the protein structure and function. It does. It marks the protein for excretion from the body. That's a major thing it does, although there are other things it can do also. And so here they've come up with this thing here showing that glutathione inhibits 3 steps in this ACE-renin ACE process, but that's not all it does. This is really very nearsighted. I mean, it basically reverses your oxidative stress. I mean, glutathione does a hell of a lot more than just these 3 sites. I mean, it does. This is a nut site, but this is the anti-inflammatory effects in this one process. And so we can ask the question, can treatment with a drug that increases intracellular glutathione help with the prevention and recovery of viral infections? And the answer is a resounding yes. And here's a paper. It was published in 2011 by some Japanese guys. Antioxidant therapy as a potential approach to severe influenza-associated complaints. And then the abstract says, these results suggest that an agent with antiviral and antioxidant activities could be a drug of choice for the treatment of patients with severe influenza-associated complications. This review updates knowledge of antioxidant therapy as a potential approach. And I'm telling you, that's what I have. That's what we worked on. And we knew this 11 years ago, and we've been trying to get this through the FDA ever since. And here's one, glutathione increased by the N-butanol glutathione derivative, which they call GSHC4, inhibits viral replication. In other words, you can take a modified glutathione molecule that isn't, that you can deliver to cells in culture or have mice drink it, and it will prevent the replication of the influenza virus. And that's all listed in this abstract where you see where the red R's are. It said the treatment with GSHC4 increased glutathione content in organs, reduced viral replication, and induced a predominant Th1 response. Now, why aren't they using this on the COVID patients right now? This is, I'm trying to think when this was published. No, I haven't. I should have it on there, the dates that was. I can't find it, but this is quite old. I mean, this is not brand new. So the antioxidant concept, helping to maintain a healthy total and reduce glutathione level. It is of major concern that toxicities in sporadic or chronic illnesses or infections can reduce total or reduce glutathione for such a period of time that recovery is made difficult because of the buildup of many toxins that require a consistent higher level of glutathione for effective removal. In other words, if you're exposed to mercury and you're healthy, you will probably excrete that mercury very rapidly, or was within a reasonable period. But if you have the flu and you're sick and your glutathione levels are in the toilet and you get exposed to mercury, that mercury is going to stay there a long time and get deeper in the body and interfere by binding with other proteins, and it's going to have a problem. If a significant percentage of mitochondrial or dysfunction or damage, they can continuously catalytically produce hydroxy-free radicals at a rate that prevents glutathione recovery to a normal level. And the example of this would be radiation sickness. When you get hit with radiation, it destroys your mitochondria. Your mitochondria do not work effectively. And if your mitochondria aren't working effectively, you don't make glutathione. And if you don't make glutathione, you'll die just like if you have Tylenol toxicity. I mean, your mitochondria have to be functional, and you have to be making the energy, and you have to be making other components such as glutathione that are supported by this. Both medical, dental, and dietary approaches are needed to help the body maintain a healthy glutathione level. That's an understatement. I mean, I think it's something that we really need to address more. What is the effect of what we eat on our ability to keep our glutathione levels high? I mean, it's one thing to produce a lot of fat and a lot of carbohydrate and protein, but how do you protect yourself from viral infections if you're not eating such that your glutathione levels are maintained? Because that requires a certain percentage of cysteine, glycine, and glutamate in your diet. Whey protein is a good protein to take if you want to do that. And it says, in my opinion, it's quite unlikely that any chelator can remove mercury bound to the iron sulfur centers in the mitochondria. I mean, that is about as tight a binding of mercury as you'll ever get. And that's the reason when you are mercury toxic, your mitochondria stop working. You stop making ATP and you go into chronic fatigue. And you cannot pull that off, even my chelator. I don't think you would pull it off of that, because those iron sulfur centers are just set in a position that they would bind mercury incredibly tight, and they stop the electron transport. When the electron's going down the electron transport chain and it hits iron, the iron moves it around and it gets exchanged and travels on. If it hits mercury, it stays there and it's blocked. And that's with other toxic metals also. So you have to have the right metal to do electron transfer. Iron will do it. Mercury, cadmium, and lead won't. But they'll bind to the sulfur very tightly, and they're very toxic compounds in that regard. So just for the chemistry walks in the room, here's this: what are the reactive oxygen species? And if you look at that top reaction, the oxygen there, the superoxide anion production is the first step. You breathe in oxygen. Your hemoglobin comes to the cell, releases the oxygen, it goes into your mitochondria, and it sits there and it's waiting to be attached to an electron, which is coming down the electron transport chain. And if it leaks out, then you get a superoxide anion, the O2 minus. And then if you look at the rest of the chemistry, and if you want to study this in detail, you can see that you get down to this last one. If you have free iron, iron plus hydrogen peroxide produces the hydroxy radical through the Fenton reaction on the last line there. And that hydroxy radical is what damages and kills. This is when you say you're sick, and they say, and oxidative stress is an associated function. No, that's what's killing you. I mean, the oxidative stress is what's causing your tissues to fall apart. Hydroxy radicals, they're the actual blade of the knife that goes into your body. I mean, when you have all kinds of illnesses and it produces oxidative stress, it is the oxidative stress that kills you. The disease just produces the hydroxy radical. Excuse me. propose that increasing, and by the way, I'm CEO of the company that's developing this. A lot of the people in this audience are my early investors, and I want to thank all of you for trusting in me, because this is working. It's working. We're getting there. We propose that increasing the body's intracellular reduced glutathione levels with the Mercury would increase the body's defense against almost all viral infections, including COVID-19. Now, to be fair, the National Academy of Science Catalyst Research Group didn't agree with that. I mean, they thought there were other things more important. And they may be right. I mean, I didn't read all the grants. But NBMI's antioxidant scavenging activity, decreasing the level of existing hydroxy-free radicals produced in mitochondria, salvages glutathione, reduced glutathione. If you take it out with NBMI, and it binds hydroxy-free radicals even better than glutathione. It scavenges them and renders them no longer toxic. I mean, they're removed from the system. That's one of the things about the compound. When you say taking it, how long should you take it? You know, it chelates the mercury and the lead and the toxic metals in your body within a day. I mean, we've done that with rats. We inject rats with huge amounts of multiple lethal doses of mercury and wait until they're in convulsions, and we inject the compound on the other side, one dose, one time, and they totally survive. I mean, they survive very well. So it binds it within a day. If you don't do it, they're dead within 3 to 6 hours. And all of them were alive 48 hours later. So it had to scavenge those hydroxy-free radicals or those toxins almost immediately. And the other thing that NBMI does is a chelator. It binds mercury thermodynamically irreversible. It takes that mercury out of the system. If you add mercury, we can take and make mercury NBMI complex and gavage it into rats at 2,000 milligrams per kilogram body weight, which is enough to kill 100 rats. And it's not toxic. It gets in the body, they digest it, they poop it out, and they don't get sick. So when it's bound by NBMI, that mercury is no longer ever going to react with another molecule. And with our studies we found out the same thing happens with unbound iron. We've done this with rats that are models for hemochromatosis. NBMI, if you give it to a normal, healthy rat, it will not deplete the iron in their hemoglobin or in their ferritin or in their transferrin molecules. The normal iron stays the same. It doesn't deplete it at all. But if you have unbound iron, in other words, if you've injected them with iron or had something called a disease that causes them not to be able to get rid of unbound iron, it binds that unbound iron and renders it non-toxic. We have some beautiful research done on that. So we would say that this compound has everything that you would want to do to treat oxidative stress caused by heavy metals. And this is the molecule, a simplistic thing of it. David Kennedy has made a very beautiful movie of this molecule binding in mercury and how it does it. But those two sulfurs, where you see the two blue arrows, one of them gets oxidized and picks up 3 hydroxy radicals, totally taking them out of the system. When it does that, it becomes sulfonic charge, SO3 minus, and it's excreted by the kidney. It also chelates iron, preventing the Fenton reaction, and it chelates mercury and causes mercury to be unable to inhibit other enzymes or to produce free iron, any more free iron. And here's the antioxidant possibility. You've all seen this before, I'm sure. NBMI has one of the highest hydroxy radical scavenging capacities of any molecule ever made. And it's something you can take orally, and in 2 hours it peaks in the cells of all the cells of the body, including the brain and the bone marrow. So it has everything going for it. It's one of the most potent antioxidants ever tested. And that has nothing to do with it binding iron. That just has to do with it scavenging hydroxy-free radicals. And I've put the slide up probably 50 times in my life, so you can read it. That's the reason I put it there. But the data support the fact that Amerimed is a potent scavenger of hydroxy radicals with this really high EROC score of 200 to 300,000. And we've tested in rat liver microsomes where they're producing hydroxy-free radicals. And you can see when we do that, and this was done by, again, the chemical wonks, ICP for mass spectrometry with fragmentation. And we can show that we're getting SO3 minus is the final product when it scavenges hydroxy-free radicals. Okay, here. This is getting now back to the biochemistry. In cell biology, if you take control animals, the one on the far left, those animals, we'd say those cells are not toxic. They're happy, they're eating the food we gave them. And then but if you give them NBMI, their glutathione levels, even though they're healthy, their glutathione levels in those cells almost double. So it has a very strong capacity to increase the production of reduced glutathione. Now the next one over is what happens if you add methyl mercury, like eating fish. You see that the glutathione levels drop down dramatically. That's the one with the big arrow going up. The blue, dropping from the blue on the left there, you see it comes up. But if you put NBMI, it doesn't drop at all, because NBMI binds methyl mercury very tightly, rendering it non-toxic. And if you add thimerosal, which is the preservative found in many vaccines and medicines, you see it drop down. It doesn't, our compound doesn't bind the thimerosal mercury as tightly as it binds methyl mercury, but it does bind it, and it does prevent it from having such a dramatic effect. And so we say that NBMI increases significantly the glutathione level and controls cells in the absence of any mercury toxicity. This is why I'm being hassled by people who do stem cell therapy. Stem cell therapy won't work in a person that's under heavy oxidative stress or heavy levels of heavy metal exposure, because it kills the stem cells. So you treat them with this first, and then the stem cell therapy will go better. So this compound has a lot of functional uses. Once we get it approved, and people who know how to do those things can test it and prove it. Okay, here's the data on the treatment of 6 patients, 3 young and 3 old, that we show. And this is the total reduced glutathione. By the way, in a healthy person, the glutathione levels, GSH will be 90% of the glutathione, and the GSSG will maybe be 10%. But these were people that were sick. They had either autism spectrum disorders or older people with different medical problems. But you can see when we gave them glutathione, gave them MDMI, that their glutathione levels went up dramatically. So we know it works. We've tested it into people that were sick, and we gave them this compound, and they're reduced, and they all got better, by the way. They all, according to the medical doctors. I don't know how you say that, but some of these autistic children, and autism, the reason I made this compound, to be very honest with you, to start with, was because the mothers of autistic children, the way they looked at me when they talked to me about how sad they were that their children had been destroyed by what they thought at that time was vaccine mercury. And so that's the reason I made this compound. And it worked. They started getting better. You can argue about what causes autism, but you can't argue about the fact that autistics aren't under oxidative stress. They can measure that. There's something is causing them to undergo oxidative stress. And when you get that at 2 to 3 years of age, it's not because you're out drinking or taking drugs. So what the hell did it? I don't know. But I think there should be more severe questions asked than have been asked of these people to date. But anyway, this is telling you now that we can increase the ratio of glutathione inside cells in living people, in cells in culture, in animals, and we've done a lot more than this. But you guys are going to get tired of listening to me for any period of time. So, and this is just some chemistry showing what we've talked about. That glutathione is used. This is medicine. Medicine accepts this, and they publish it, and then they read it, and then they forget it. So this is just the final part of the talk. With the presence of heavy metal chelation accompanying the high level of antioxidant properties of be an advantage for the treatment of viral infections, bacterial infections? Yes. You're not supposed to have free iron in your body. Free iron in your body is one of the most toxic metals around. Iron overload patients, you take people with thalassemia and you give them blood transfusions, and they can breathe, they can transport oxygen, but every time you give them a blood transfusion, you give them also a bolus of unbound iron. And that unbound iron starts building up in their ferritin, and it causes them to get sick and later die. And so they call it iron overload disease, thalassemia, sickle cell anemia, etc. So this is important. And this is just the front line. Importance of glutathione levels serves as a front line reducing agent, keeping all of our enzymes protected from oxidation by hydroxy-free radicals. Sometimes we get toxic, we produce too much hydroxy-free radicals, and the glutathione gets depleted, and we get sick, and we have major problems. It serves as a natural chelator for excretion of many toxic metals, including mercury, lead, and cadmium. I can tell you we're doing studies now where we're taking the kidneys of animals that have been exposed to mercury, then treated with NBMI. We grind up those tissues and we wash them with 100 millimolar glutathione. That's a huge excess. It's 5 millimolar is the highest in your body, 5 to 6 or something like that. But when you wash these mercury intoxicated kidneys with glutathione, you get a huge amount of the mercury released into the soluble aspect. When you spin down the protein, it's all bound to 2 glutathione molecules for each mercury atom. That's 6 charges, so it's very soluble, and you can measure it. But if you treat them with NBMI, after you've treated them with mercury, and you try to extract it with glutathione, it won't take nearly as much off, not even close. Because we have proven, we've done experiments showing that NBMI mercury does not give up its mercury to the most avid mercury binding protein in your body, which is called glutathione. And so that's how the compound works. It covers up the mercury, rendering it non-chemically reactive, non-toxic, and just inert. And your body slowly gets rid of it over a 30-day period through the P450 system by attaching glutathione to the aromatic ring. And then it puts charges on it and it carries it out, just like it does every hydrophobic drug that you use. So this part here, here's some stuff that shows that we can do it. Now I'm going to get to one end of it here because I want to tell you some of the things you can do, because our drug it's available only if you get the FDA to agree that you can use it. They show that the antiviral activity of glutathione, this is another paper. Again, this was published in 1995, and it was done in Italy. Almost all of the good work on viral protection based on glutathione is done in Italy. Our NIH doesn't want to do that because they like selling vaccines. And this is, again, the structure of glutathione. And if you look at that molecule, when you attach it to the sulfur, you have a carboxylate group, which would be negatively charged. At physiological pH, you have an amine, which would be NH3+. So you have 2 charges there, and you have another charge on the other end. And you have these out there that makes things soluble and makes it very recognizable to glutathione binding compounds. So, this is the last thing I want to talk about, and that's that it's called the Meister cycle. People ask me, what is the best thing to give somebody that is sick? And I didn't learn this, I got this from David Kennedy and Rich Fisher and other people on this academy that were telling me about the value of high-dose vitamin C IV. And, when I heard that, I thought, vitamin C doesn't bind mercury. Or interact with hydroxy-free radicals? How can it do that? And I found out it's called the Meister cycle. And what it is a line here, you increase with vitamin C NADPH levels, and that takes oxidized glutathione and reduces it back to 2 glutathione molecules. The IV delivery of high levels of ascorbic acid is known to fight viral infections. I've heard this all my life. Since I've been with this group anyway. And so you have to say it has to have an antiviral activity. How does it do that? It does that by increasing the intracellular level of glutathione. I don't know how well it does it compared to our compound, but I know it does it. And this is the Meister cycle, and this thing is so yellow because I've been talking about this, I think, since the first 2 or 3 meetings I talked to you guys about how reduced ascorbate, or reduced vitamin C, comes over and takes oxidized glutathione to GSH. Now, this is what I've been teaching you guys, and I want you to recognize my kindness, because here's the real Meister cycle. I thought maybe you could handle this one, but this one it's even tougher for me, but it's in here. And it says this is a glutamyl cycle proposed by Alson Meister. He's a great scientist. And it's involved with retaining your glutathione. And this is a compound that we talk about, and David Kennedy's made a film of this, binding mercury, and I suggest you all, if you get a chance, look at it, because it really defines how the compound binds. And essentially, for those of you, it has two arms, two sulfur groups that will bind to mercury. And it has two nitrogens. You look at the elbows, and their blue things are nitrogen. And nitrogen has unshared electrons in that outer part, and when it binds to a metal, it can do what I call induced fit. It can bind to metals with different coordination chemistries, because all metals don't bind things alike. Some bind it, like mercury's 180 degrees apart, iron has different angles, but this molecule can vary around, and when it does it, what we found with it, it binds almost all the toxic metals if they're attracted to sulfur. These sulfurs can move around at whatever angle the metal needs the most to make the tightest bond, and that's where it holds it, and it binds them thermodynamically irreversible. It'll take almost all these metals down to less, if you put it in 1-to-1 ratio, it'll take out over 99% measured of the metal into a pellet at the bottom of the liquid. And if you try to redissolve that in anything, you can't do it. It will not let that metal go. So it makes an inactive thing. And in your body, you don't form a precipitate because your metals aren't that concentrated. And so then your P450 system comes up, and at the very top part of that ring system, that aromatic ring, it puts a hydroxyl group there and attaches glutathione to that. So this molecule, when it binds to metals, is totally uncharged. It's not water soluble, it's not soluble in anything. But when you attach a glutathione to the top part, it now has 3 charges to it, makes it water soluble, and you move it out of the body. It goes slow, but it goes out. So this is a summary, and I'm all done. Okay. You can read this as well as I can read it to you. Okay, so. And thank you all for this, and I would be happy to answer any questions if you want. Anybody has a question you want to ask? We're setting up a meeting right now. The FDA made us do what they call a pivotal efficacy study. They put us onto the animal world because they agreed that this compound is not toxic. We couldn't give enough to make an animal really die or get sick. I mean, especially at the level that you need for chelation. But we had to take rats, and we had to inject them slowly with mercury at low levels to make their kidneys go bad, but not so bad that they died. And they had to last at least 5 days. So we got them up to 7 days. Their kidneys are shot, and then we had to put them on NBMI and show that we could cure them. And we've got that done. We've submitted that. We're submitting that to the FDA now, and then we'll have we did it on 6 rats per group. The data is beautiful. It's as good as you expect to get in that kind of a study. And so now we have a meeting with them, and we ask them, what do you want us to do now? We know it's not toxic. I mean, you're putting out so many toxic drugs. This should make you feel good right there. And and you know, we've done the studies where we gave them lethal doses of mercury and kept them from dying with this drug. It should be approved just for that reason, for somebody that, you know, inadvertently gets exposed to lethal levels of mercury. So we're meeting with them, and when we meet with them, they'll tell us how much time we have left to go. But we've got the CMC, the chemical manufacturing compliance, all done. We've made the engineering batch that they require to make, and then this next month we'll make the 3, they have a name for it, 3 batches that we can now put into capsules and give to people, but then they make us wait a year to make sure it doesn't break down in storage. We've already done that with our compound. It has a 5-year shelf life right now. I mean, at the end of 5 years, there's no change. So at the end of 10 years, there's no change either, I can tell you that. But we have to do what the FDA says. They're the 800-pound gorilla. So I say we're at the point where the FDA has to tell us what to do. But we've done everything. The compound is efficacious, it works, and it does a lot more. We have finished, we're in the process of finishing 2 phase 2 studies. We finished the treatment of mercury toxic miners in Ecuador. We didn't have a single drug adverse effect, and we had a lot of positive changes. Showed the decrease in the mercury going out in the urines, indicating our compound was binding the mercury in their bodies. We did a study on COPD patients, 10 of them, that were in hospice. In other words, they're in bad shape. NBNI will not remake lung tissue that's been destroyed. But if you take it early enough, it stops the lung inflammation. And we've had really good results with people in the early stages of COPD taking it, smoker's cough, that level. It just takes it away because it binds, it stops the inflammation. And you still have lungs to breathe with. And so we're now finished close to finishing 2 other phase 2 studies, one on thalassemia, iron overload, and we didn't have a single adverse effect giving 600 milligrams a day to these people for 28 days. And none of them died, and I don't want to say what. Let me tell you, I'm very happy with the results. Okay, it did a lot better than everything else they have out there that they're using now that cause people to die. And we did one on a study, again, on atypical Parkinson's. And the reason we did it on atypical Parkinson's, we started out to do 10 patients. And after about 6 or 7 days, they asked us, would we consider tripling the number of patients and doubling the length of time we treat them and double the dosage we give them, because they were seeing positive effects in a disease that has no cure, a lethal disease that has no cure. But I don't have the report on that yet, so I can't say more, except things are looking good for us. But if you think that that's going to have an effect on the FDA, you're wrong. I don't know where they come from. I can just tell you that they are not proactive in finding medicines that the people need. They're just not. Yeah? Yeah, we have done several comparisons with N-acetylcysteine, and it works. But it doesn't work nearly as well in any of these things as Imeramed. I'm sorry, John, but I'm not hearing you.
How about comparing the NAC to just using cysteine orally?
Well, we haven't done it because it's not, but we've done it in animal models and cell models, and there just isn't the capacity. N-acetylcysteine does not bind and remove mercury. It's not got chelation properties. It's not got one sulfur on it. It can bind to it, but it doesn't cause it to go down. So I don't. That's a good question, but I don't have the answer to it. Except when we've compared. Where we've added iron and copper to lung lavage samples, iron and copper, and so it increases the oxidative stress, and then we add Imeramed, DTPA, EDTA, etc., and to see which one slows down the oxidative stress buildup, and nothing touches NBMI except DTP, and it's a very lethal chelator. It's sort of like a bad iron chelator.
Do you know why the usual treatment of Tylenol toxicity is inhaled NAC versus taking it orally or taking cysteine itself?
I don't know. I'm hoping somebody will, when we get it approved, will try that, work on that. But that's not a level of expertise I have, John. But that's a great idea. Yeah. Get the microphone. I don't have the best of hearing, folks.
Yeah, I was going to say, if we have 5 more minutes, so.
There's extensive literature about metabolic syndrome through a man named Resnick and another group named Wiglicki that shows that you cannot make glutathione if you have magnesium deficiency. And this is a very widespread deficiency in the modern world. So when you use cysteine, you're trying to make glutathione. Well, you can't make glutathione if you don't have all these cofactors, so that's another problem.
Mm-hmm. Okay, thanks. Thanks, Bob.